All right, welcome back to the second part of our arthropod lecture. So, just a reminder here that we're going to be looking at the characteristics of arthropods. Last video we covered the spiders Chelicerata and the Trilobata, so the extinct trilobites, as well as the reasons why arthropods were very successful. So, if you missed that, go back and check out part one. So in this video, part two, we're going to be looking at the last four major groups of arthropods and describing their features within. So the first major group is Mirrorpods. Mirrorpods are terrestrial. These are your centipedes and your millipedes. Millipedes eat decaying leaves and dead matter, so they're not going to necessarily can hurt you, even though personally I don't really like the way that they look or feel. Millipedes have two pair of segments per legs, while centipedes are carnivores or carnivorous, and they only have one pair. And so looking at the prefix centi and milli, so centi being 100 and milli being 1,000, they don't really have 100 or 1,000 legs, but it is a good way to remember which one has the most legs per segment. So looking here at a centipede, very stereotypical here. You'll find these in rotten logs, typically looking for some type of beetle larvae or beetle eggs. Again, they are carnivores there. These actually a lot of these can hurt. They can bite. Some do have venom, which can make a little irritant there. Millipedes up here in the top left are, like I said, personally, I just don't like the way they're usually in rotten wood and leaves and stuff as well. But they're not. They can't hurt you. They can ball. They'll kind of ball up in a little tight U-shaped pattern, but no, they're harmless. So that moves us to the second major subphylum here, Hexapoda, and these include insects and all their relatives. So these are terrestrial and in fresh water. There are some that live solely in fresh water, and we're going to look at the internal anatomy of the grasshopper as our exemplar. So looking at the head. So you'll have a couple quiz questions on the external anatomy, internal anatomy of a grasshopper. Looking at the head region, you can see, hide this, that there are a lot of structures here that one of the reasons why their advancements over segmented worms is that each of their segments is highly specialized. So again, you have just kind of orient yourself around here. You have ocelli, these little simple eyes, kind of see light and dark. And then you have the compound eyes, which are going to have the ommatidia. So again, looking through lots of straws there. The forehead region being the vertex. Don't really worry about this little cleavage here. We're not going to concern ourselves too much in the weeds. Antenna, of course. The frons, the genia of the cheeks there. The clypeus, mandibles, and of course the labium. So again, the genia of the cheeks, the frons, labium, so lower jaw. We'll look at a different view here from the side and expand these mouth parts so you can kind of see this. So again, the labium is the lower jaw, maxillae with the maxillae palp, hypopharynx there, so that's kind of like the tongue region, mandible, and labrum. So most people will kind of confuse labrum and labium. So labrum is upper, so the upper lip, and labium is the lower jaw. So looking at the body of this grasshopper here, we see a lot of things in this that most of our insects relatives have. So it's basically broken up in three parts, which you may have learned earlier in your elementary training: head, thorax, and abdomen. But so the head, again, we just looked at lots of structures inside the head. The thorax is really broken into three different groups: prothorax, meso, and metathorax. And so the mesothorax is typically where the prothorax is where you're going to have a lot of your flight muscles. The mesothorax is also where your flight muscles will attach to your wings there. And then on the abdomen, you'll see something called an ovipositor if it's a female. Ovipositors typically, transition to a laser pointer again, typically will have this stereotypical prongs coming out of their abdomen that kind of gives it away. So really for the first time in the animal kingdom, we're able to externally tell the difference, in some insects, but not all, the difference between male and female. even without, microscope or really knowing what you're looking for. So again, we got the forewing and the hind wings. We got two pair of wings here. Not all beetles have wings, or not all insects in general have wings. Some have like the beetles there. The forewings are hardened into what we call an elytra, and then some can fly by the high wings, some do not. So flight has evolved multiple times throughout the Insecta class there. Interesting thing here, we've got again three pair of legs, so 6 legs total. In this case, we got 2 pair of walking legs and then 1 large pair of jumping legs, which obviously we know from our grasshoppers here. And then, interestingly enough, grasshoppers have something called a tympanium. So if you're a percussionist or have been in some type of band, you may are aware of a drum called a timpani. And this essentially is the same type of concept where we have essentially a structure with some type of membrane or sheath surrounding it there, and that's going to be how this insect is able to hear and also to make sound. Not shown here. Trying to show here are these spiracles there. Looking through, make sure I got everything. Perfect. So again, know your external anatomy of the head as well as the anatomy. Let's look at keep moving with the parts of the leg. So lots of anatomy here to learn. Luckily, though, if you've had A&P or you know your bones at all, you'll see a lot of the same names. So starting with essentially where the jumping leg attaches to would be our hip, is the coxa. The long bone is the femur. Separating again because they are segmented. A slight movable joint here is the trochanter. So coxa, trochanter, femur. Then the femur connects to the tibia, which often will have these tibial spurs, tarsus, and then finally these claws. So again, you want to make sure that you know the anatomy of the head, external anatomy of the body, and the external anatomy of the leg. Now we'll move inside. We get to see a lot of different structures and ideas. So again, these guys do have a brain, right? So surrounded by a nerve cord. What's very interesting is that this nerve cord continues to wrap around the intestines, and the nerve cord is ventral. So we still have this ventral nerve cord. We have the crop and we have a gizzard, dorsal artery with the heart being in the back, in the abdomen. That's kind of different, right? We have the ovaries, because again, we do have an ovipositor here, with the vagina, the anus. Malpighian tubules are essentially the way that these insects regulate metabolic waste. And then the gastric ceca here, which isn't labeled, but will be here in a few minutes. So, as I mentioned before, insects have diversified over several times this idea of flight. And especially when we start to see the adaptation of gymnosperms, which are stereotypically like your pine trees, and your pine trees are your best example, and the expansion of your angiosperms, which are essentially all of your flowering plants, not just trees, but plant flowers as well. And insect and plant diversity declined during the Crustacean extinction, but has increased in the 65 million years old. So we basically saw, like most things at fifth mass extinction, the KT boundary, lots and lots of animal life in general saw a large dip in number, with many lineages of insects and animals dying off, going extinct at the KT extinction when the dinosaurs were died. But because they were able to, they had lots of genetic diversity and lots of different ways to feed, lots of insects diversified. And when the evolution of plants, or especially flowering plants, develop, we see a large tick in insect, specifically beetle diversity, help pushing the numbers well past. Right now, we know for sure a little over 400,000 beetles alone with names. To put that into perspective, there's a little over 5,400 animals, give or take 20 or 30 or so, mammals today. And just beetles alone, not just all insects, but just beetles, 400,000. So remarkable, incredible diversity and success that this lineages have been able to find. And one of those major regions, as we discussed before, is flight. And so flight has just so much more than just being able to escape predators, but now I can move. If food is bad, I can move. If new guys come in and are better able to outcompete for me, right? Because competition is one of the major factors in our evolutionary ideas. Then I can either, like I said, you really have three options. I can adapt to eat a new, different type of plant. I can move, or I will die. So if I have the ability to fly, I can find new habitats much more quickly than if I was forced to crawl or wait on some type of other animal to ingest me, then later poop me out, and I've come in a new environment like a lot of our worms did early on. So again, just a lady beetle here in flight. Great picture. Another one of the key aspects of diversity there that's really helped their numbers is this idea of metamorphosis, which we talked about in the first video. You have complete and you have incomplete. So in the incomplete metamorphosis, the young are called nymphs. So these are perfect example is your grasshoppers. And so essentially here's our adult. They lay their eggs, they hatch, and then we have these nymph or instar or larval stages, right? So first nymph stage, second nymph stage, third nymph stage. And you remember one of the characteristics of this arthropoda phylum is this idea that I have to molt. I have to shed my skin, exoskeleton, every time that I grow. And so in the case of this exemplar here, we've got 1, 2, 3, 4, 5 nymph stages before I'm finally an adult. Complete metamorphosis is what we're probably very familiar with, especially in our second and third science grade classes, where we go from a egg to a larva to a chrysalis to a butterfly. And we talked about the advantages and disadvantages of that. One of those advantages is, again, if you look at just the morphology of the larvae compared to the adult. And so the larvae is well adapted to crawling on top of plants, underneath plants, and those little mandibles there are phenomenal at chewing and tearing and ripping and shredding plant material. But if you look at the mouth parts of the butterfly, the mouth parts have been converted into what we call a proboscis on its rostrum. And so this proboscis, in this case, is kind of like one of those birthday kazoos, where when I put pressure, the kazoo will force out. And these are essentially the same thing. So I land on a flower, I blow, muscles control the contraction and expanding of this proboscis, and I'm able to essentially suck nectar from my plant. So now I'm going from flowering plants, specifically the sugar, to the larvae, where I'm eating the solid plant-like material. Most insects here are separate, right? They're separate male and females. So again, they're dioecious. They reproduce. So again, and even with these guys here, they're able to tell a lot of, or especially if you're a trained morphologist, you can tell externally male or female, which to me is just crazy to think about, that I can take an insect for the very first time in our animal evolution, to think about that we have external male and female genitalia, or at least sexually dimorphism there. Not necessarily that the genitalia itself is external, like it is in mammals for instance. They're able to find each other by their sound. So think about grasshoppers. Sight, so your lightning bugs, for example. So your glowworms, your females are your glowworms, and they're sitting there on the ground, and they're producing this pattern that the males will sit there and recognize and then fly toward it. Also odors, so your pheromones. Insects are incredible beneficial as pollinators. It's estimated that if, for example, bees or other types of insects were to go extinct, that we would lose billions and billions of dollars to the world GDP because of lost crops, right? But not only are there some great insects, there's also some really bad insects that are causing billions and billions of dollars in damage. One major concern right now is the Asian longhorn beetle. That's a generalist feeder on any hardwood. So some other insects that cause major damage, like the pine borer, the ash beetle, those are very specific to those types of trees. But the Asian longhorn beetle is one that's really has a lot of concern in the U.S. here, because it's such a generalist on a hardwood And it's estimated that if it does break quarantine from the 12 or 13 different locations that it's been found, the economic impact on our hardwood forest is just devastating, catastrophic. Depending on what type of systematic study you follow, there are anywhere from 20 to 30 plus orders of insects. So this insect phylogeny is always changing as we're acquiring mostly new phylogenetic techniques are in DNA and in genomics. So we're sequencing partial to whole genomes and trying to put these back together and figure out who's related to who. To the traditional of morphological, where we're looking at larval and adult characteristics and trying to figure out, again, that family, who's related to who. So some textbooks will say that there's 30, some will say there's less than that. So again, it's always in flux and depends on there. But I just want you to know 4 or 5 of these major orders here. Coleoptera being the beetles, Diptera, which are your flies, Hymenoptera, your bees and wasps and ants, Lepidoptera, your butterflies, right? So just these 4 major orders here. And the reason for that is because these 4 orders make up most of the diversity within the insects. So again, with the Coleoptera, here's a little weevil there, over 400,000 species. Diptera, all your flies, 150,000. Hymenoptera, bees, wasps, and ants, 125,000. Lepidoptera, over 120,000 species of moths and butterflies. Interesting fact, just totally free here, Hemiptera are known as true bugs, so it's really incorrect to say when you point to an insect, call it a bug. All right, so all hemipterans are true bugs, but not every insect is necessarily classified as a bug. So just interesting tidbit for you. So let's look at the digestive system of the grasshopper. It's incredibly complex and fun to study. So again, these mouth parts are highly modified, like a lot of our insects are, their segments are, for chewing and cutting plant material. So the labrum and labium, the upper and lower lips, are functioning there, and they'll hold the food in place so that the mandibles and maxillae can cut it. So essentially we have almost, again, our upper and lower jaw. So our labrum would be upper jaw, labium will be lower jaw, and then as we're holding that food in place, we essentially have shears that can sit there and grind and cut the plant material away. Food is moistened by saliva, just like it does in our mouths, passes through the esophagus, and goes into a crop for temporary storage. So these guys have both a crop and a gizzard. So the crop holds it. The gizzard is where all of your grinding and shredding is going to be before it's pushed into the midgut. And the midgut's where a lot of your enzymes are going to be released. And so now we have mechanical digestion. Now we've got chemical digestion. And so these enzymes are going to break down the food, larger food components into subcellular components that the cells are then able to absorb. And then any undigested material is going to go through the hindgut. And it's not necessarily that the grasshopper couldn't digest its meal, but it goes through the system so fast that not all of the nutrients get absorbed. And so this is going to be solid material that's going to be pooped or insect cast, right? So looking at the internal anatomy here, decent picture here. So again, with anatomy, figure something out, a structure that you can always find. Personally, I like to do the pathway of digestion, because I'm going to usually get 5 to 7 structures in an internal anatomy diagram. So starting with the mouth, go through the pharynx and esophagus, just like we did in the worm. Mouth, pharynx, esophagus. Crop, you can see how large that is. We have a little bit of a squeeze into the gizzard. So the gizzard is covered by what's known as the gastric ceca. The gastric ceca there is what's helping essentially the midgut. That's where where your enzymes are being made to help digest chemically what's in the gizzard. So crop, gizzards, again, underneath the gastric ceca there. We got a little bit of another squeeze, the pyloric valve, just like in our stomach, which is kind of cool, before we go into the intestines. And finally, the rectum. So they have a little bit of distinction in this diagram between ileum and rectum. So ileum, again, is where a lot of your nutrients are going to be absorbed throughout and travel throughout the cells and the body. And then undigested material will go out the rectum and the anus. So let's go through that one more time. The pathway of digestion: mouth, pharynx, esophagus, crop, gizzard. Chemical digestion by the aid of the gastric ceca. We're going to kind of holding spot this pyloric cecal to the ileum or intestines, rectum, and anus. Okay. So again, three major regions: foregut, midgut, hindgut. Malpighian tubules shown here again are for metabolic and excretory waste there. Don't worry about the ventriculus there. Did I get everything? Looks like I did. So again, you'll want to know this pathway as well as this diagram because I usually show this on quizzes and exams. All right, the circulatory system. So nutrients are transported through the body. This is an open circulatory system. So just like we saw in some of our mollusks, in our clam, for example, so heart is going to pump and then blood is going to travel through sinuses and get connected by an additional vein going back to the heart. So this blood is hemolymph instead of hemoglobin. And so in hemoglobin, like in our blood, the major component that's holding on to oxygen is the iron molecule, which is why blood tends to be more red in color. In the case of insects, it's a copper component, and so in essence, that's why their blood is more of a bluish color. The heart there is located in the abdomen, and the thorax pumps the hemolymph toward the head, where again it moves through those sinuses into this body cavity called, again, a coelom. A coelom is just a type of body cavity. So the hemolymph then slowly makes its way back to the heart to be recirculated again. So again, in regards to stamina and major burst, grasshoppers and insects really just don't have it. So if you've ever tried to catch grasshoppers or chase a grasshopper, you're going to see that it jumps really quickly. It can kind of fly, fall with style if you're a Toy Story fan, then try to hide as best it can before being forced to jump and fly again. So again, it's not going to have long, sustained flight like some insects can, for example, like the monarch butterfly. So again, this is just a diagram of the circulatory system in a grasshopper. So again, just showing you the difference in open and closed circulatory systems, right? The nervous system. So the nervous system, these guys do have a brain. And when you dissect these, you're not going to be able to see the brain, at least with a little, 4 or even 10 times magnification hand lens that we have. But it is followed by a ventral nerve cord. So again, the nervous or the spinal cord essentially is on the belly side. Ganglia there, just small nerve centers that can help control each segment and boost signals from the brain. So just kind of like in our nervous system with these type of receptors, is that each segment has ability to integrate and sense different senses. Send that signal to the ganglia. The ganglia then kind of connects that, calculates, is it important enough to send the brain? If it is, it's going to send that to the brain, and then the brain can integrate that, figure out if it's worth a overall body response or not. So there's lots of fine control within each ganglion segment there. So in the head, the nerve extends from the brain to the sensory antennas, sensory organs such as antenna for touch and smell. The simple eyes, I remember those little 3 eyes in the middle of the forehead. Those are going to measure light intensity. And then the compound eyes, composed of hundreds of lenses, that's that ommatidia, provide a wide field of vision and help detect motion. So these guys here are incredibly adept at knowing where you are at all time, seeing the difference in light and dark, and then also being able to detect the motion that goes along with it. So The abdominal ganglia connects to a sound-sensing organ called that tympanium. Again, this is specifically in the grasshopper there. This oval window on the first abdominal segment, which allows hollow and airflow there. So the sounds cause the tympanium to vibrate, and just like in our ears there. So as sound is coming through the speaker here now, the frequency of the sound is hitting your ears and causing little hairs in your ears to move. That movement there is hitting your tympanium, your eardrum, and your eardrum is then causing action potentials in your inner ear to register as sound. And then the amazing brain process that where your brain interprets those frequencies of action potentials into language is just mind-blowing. It's awesome. So that concludes the insects there. And let's look at briefly the crustaceans. So these crustaceans, subphylum Crustacea here, crusta means shell. So these are your crayfish, your lobsters, your shrimp. These are going to be mostly marine. There are a few like freshwater shrimp. So there are a few in crawdads that are freshwater. There are some that are even terrestrial. But again, most of these are found in salt water. Right. So we have a couple major structures here. These guys have two different types of antennae. They have antenna, which is a long antennae, and then they have antennules. So two pair of antennae. Great diagram here of the external anatomy, again, showing you the long antennae, the short antennules. Moving forward, we have a simple eye. We have a cheiliped, so what most people call the pinchers, at least around here, actually called a cheiliped. And then we have the carapace, which is the shell. And then we have something called a supraorbital spine. So let's break this word supraorbital. Orbital is typically in reference to or around the eye, right? And then supra means on top. So literally an eye or a point spine on top of the eye. So the carapace and the here make up the cephalothorax. So from the head to the carapace. So head and carapace make up the cephalothorax. The abdomen made up of lots of little segments here. And then we have these little swimmerets on the bottom, which we flip this over in a live crawfish. You'll be able to see this a lot better. 4 pairs of walking legs, which control the gills. So as you'll see when you dissect this, you move these walking legs back and forth, you're going to see the gill, which is just underneath this carapace, begin to move back and forth. So essentially, these guys have to walk to get oxygen to flow through their gills, unless they're in a current. So Teslan, Mirrorpods, again, we'll flip this over so you can see that. So the abdominal somites, 2, 3, and 4, swimmerets. This is how you can tell if you're a male or female. We'll flip this over and you'll be able to see that again a little better. Cephalothorax, the rostrum, antennae, antennules, the cheiliped. Again, highly diversified and specialized for mouth parts. So we don't get too crazy in this. You can dissect these. I do have a dissection sheet where you kind of pull these off and kind of match where these go in what they're formed. But again, you can kind of see there the walking legs, mandibles, the maxillae, the gill with a little baler there. Some crabs, such as this, have a large modified cheiliped for again, usually for displaying some dominance type behavior, as well as this. Internally, though, it gets even stranger to me, is that you have two different stomachs. You have a pyloric stomach and you have a cardiac stomach. Within the cardiac stomach, so this is what you're looking at now, inside the stomach, there are 3 small teeth called the gastric meal. So the crayfish or the crawdad will sit there and eat, go down his esophagus, and it's going to go to this cardiac stomach. And it's inside this cardiac stomach, you have these little 3 teeth that are going to begin to mechanically crush this food before passing it on to the pyloric stomach. The crayfish also has a liver. Just like our liver is going to produce enzymes to digest before going through the mid-gut there. So again, the cardiac stomach, the pyloric stomach, I got a better picture coming up here shortly. Most of these are nocturnal. So again, the pathway of digestion, mouth, esophagus, cardiac stomach, pyloric stomach, midgut, intestines, anus. So again, a great way to get lots of the internal structure is again to follow the pathway of digestion. Circulatory system is similar to the worm, but it is open, so no closed veins there. We have heart. We have 7 arteries. Again, the sinuses, which are going to collect the blood and turn it back into the veins, back to the heart. We got a large coelom around the pericardial sinus, so around the heart. So the blood, again, is nearly colorless because it contains that copper component. Interesting, though, it can clot, which is a nice factor. So we do have some thrombocytes, so some type of platelet structures that can help clot there. The gills are extremely prominent during a dissection. So here is, we've taken the carapace and removed it. We just cut along the dorsal side and then kind of peel this off. And you definitely can see the gill-like structures there. You move these legs, and these gills will also move. So again, moving the carapace on the dorsal side here. Digestive gland is fairly large. The heart, so we've taken away some of the digestive gland, moved it away so you can see the heart there. Spread out the gills so you can see that. They're even indicating here an ostium, an opening for the ear, so this crawfish can hear sound. The excretory system is done by a green gland, which is similar to the kidney. But interesting enough, the kidney is by the brain and the eye. So the green gland consists of this portion, essentially a thin, like bladder, and then will pee through this renal pore that opens just behind the eye. So essentially, these guys will urinate right behind the eye, which is just a remarkable adaptation. If you think about it, and why not, like most organisms, either pee through their skin, which makes it more difficult because of the exoskeleton, or out around their genitopore anus. It seems strange. So the difference in crayfish and lobsters, typically just the size, the same order but different families. Lobsters are so much bigger. Again, in Tennessee, we don't have lobsters. We're landlocked. Lobsters are essentially all ocean, marine, while crayfish can live in creeks, ditches, lakes. I mean, we're covered up with crayfish around here, especially during the springtime and when the Mississippi River floods. So dorsal view again, the ventral view. So if the swimmerets 1 and 2 make a V, and it's very rigid, and this V is anyway, because a lot of times when you get these specimens that have been in some type of dissecting fluid, preservative fluid, that all these swimmerets are kind of in this V form. But if the first and second swimmeret have a V and have this little bitty, almost like ball-like tip at the top, you have a male. The females, typically, they may have, again, it may be pointing up, but it's a lot more fluid. It's a lot more plastic in nature than there. So opening of the green gland, we have the tesla and the Uripod. So the Uripod is the actual external part of the tail. The tesla is the interior part. So again, a great job here of showing the internal part. So again, these are the gills, very large structure. The digestive gland, which again normally makes up a lot of this structure, but it's been removed so you can see better. Cardiac stomach, there's this little ridge that kind of gives it away of which stomach is which. Cardiac, then pyloric, then the intestines. Compound eye, just below the compound eye is this green gland, and it's not really green, so don't let the name fool you that you're looking for. If you're dissecting this, looking for something, looking for a little green gland, it's not green. So again, don't let that fool you. So that concludes Arthropods video 2. So again, hopefully what you picked out was the major characteristics of arthropods, why they are so successful, why we have so many. Again, we gave 6 different reasons in video 1 why they were successful. And then the last 2 videos, or the last video looked at 4 major structures, Mirrorpods, arthropods, arthropods are the whole thing, Insecta, and the crayfish. My mind just went blank. Back here. Crustaceans. Wow, that was a very difficult one to remember, right? Crustaceans. And then the first two videos, we looked at the trilobites and the Chelicerata. So again, hope you enjoyed this. Hope this makes some sense to you. And look forward to the next video on starfish. So we will see you soon.